Now, the complement system describes over 20 proteins which are found in the blood. They're produced by the liver and they circulate around in inactive form. They are proenzymes. So most of the time they're not doing anything. But they have the potential to be inflammatory mediators and to be antimicrobial, antibacterial substances. And complement was actually named because it complements the inflammatory process, because it can act as they act as inflammatory mediators. An inflammatory mediator is something which brings about or facilitates or enhances the inflammatory process. So when complement proteins are activated, they enhance inflammation and they are directly antimicrobial. But most of the time they're not doing anything, they're just circulating in inactive form. So what triggers them off? Well, fortunately, bacterial cell walls are chemically different to the cell walls in our body. And the bacterial cell walls contain bacterial polysaccharides, long chains of sugar molecules, polysaccharides. And these are unique to bacteria. So when these are detected, the body knows that there's bacterial infection present, and that triggers off C1, the first complement chemical. And when C1's triggered, that triggers C2, which triggers C3, which triggers C4. It's a cascade, a complement cascade. So it's triggered off by the direct presence of bacteria, but it's also triggered off by the presence of antibody-antigen complexes. So it really can be part of the innate immune system because it can be triggered by bacteria straight away when we haven't previously been exposed to the bacteria. Or it can also be triggered to infections that we already recognise because there's already the infection, the antigen, and the antibody which the immune system has previously made. So it can be triggered off in either of those two ways. And activated complement is an inflammatory mediator. It's facilitating the inflammatory process. And it also forms a very interesting substance called membrane attack complex. Basically, it forms hollow tubes. And these hollow tubes perforate the cell walls of the bacteria. So complement, several molecules of complement can form like hollow tubes. And these hollow tubes will be inserted into bacterial cell walls. So we have these hollow tubes in bacterial cell walls. And what this means is that there's now a communication between the tissue fluids and the contents of the bacterial cell. And mostly what happens is this lets water and salts in, and the bacteria will swell up and pop, and it will die. And here's a diagram I made before on this. Holes made in a cell membrane of a bacteria by this membrane attack protein or membrane attack complex. And the membrane attack complex is actually made up of several different complements all coming together to form these tubes which punch holes in the bacterial cell walls, killing them. So, I've got a large blue bacteria here that's causing infection. And we need to get rid of it. Because if we don't, this will multiply every 20 minutes and we'll have a rip-roaring infection that's potentially life-threatening. So here I've got a hollow tube. Let's see what happens. Wait. Yep, that worked. It killed it. There you go. That's the way that that's the way that complement works. So just to review the functions of complement, it enhances or complements the inflammatory process. As part of that, it attracts phagocytes, the macrophages and the neutrophils, to phagocytose infection and dead body cells. It can also act as an opsonin. Remember from phagocytosis, these opsonins, under the process of opsonization, label material that needs to be phagocytosed. Then the phagocytes come along and do it. Opsonins, if you like, prepare the food for the phagocytes table. Opsonin actually comes from a Greek word meaning cooked meat, it's ready to eat. So when something's opsonized, it's ready and labelled for the phagocytes to dispose of. And as we've just seen dramatically demonstrated, complement forms the membrane attack complex, therefore facilitates cyto means cell, lysis means to break up. Cytolysis and therefore death of bacterial cells.